Okay, hello and welcome to everyone joining us here today. Our talk is entitled Calcium Imaging Done Right, From Theory to Practice with Illustrations on Nautilai. My name is Ari, and I'll be helping guide us through this event. This webinar is designed to be interactive, so if you'd all like to go ahead and let us know in the chat where in the world you're tuning in from right now, that would be a great start. Additionally, the webinar does include a Q&A session, so please feel free to submit questions for our speaker anytime throughout the presentation. For those unfamiliar, you can find the Q&A and the chat functions at the bottom of your Zoom screen, and we'll attend to those questions after the main presentation. Now it's my pleasure to introduce our speaker for this event, Dr. Shane McMahon. To give a brief background, Dr. McMahon is a product manager here at Curi Bio. He graduated from the University of Wisconsin in mathematics and physics and went on to complete his graduate studies in biophysics at the University of Wisconsin, where he focused on developing methods for quantitative calcium imaging. He completed postdoctoral fellowships in the Department of Neurosurgery at the University of Bonn, Germany, where he focused on developing methods for investigating neurotransmission, and the Department of Neuroscience at the University of Wisconsin, where he focused on designing and building optical instruments. Dr. McMahon joined Curi Bio to leverage his experience in fluorescence imaging to lead development of the Nautilai imaging platform. Now, he plays a pivotal role in defining strategic planning and development initiatives for Curi Bio's instrument platforms. Okay, without further ado, I'd like to hand over the presentation to our speaker. Shane, please begin whenever you are ready.
All right. Thank you for that introduction, Ari. And thank you to everyone in the audience for attending this talk titled Calcium Imaging Done Right, From Theory to Practice with Illustrations on Nautilai. Today, we'll cover an overview of our 3D tissue platform. We'll discuss key considerations when creating your calcium imaging protocols. And then I'll share some example data that we acquired on our Nautilai instrument. But first, I'd like to give a brief introduction to Curi Bio. Curi Bio was founded in 2015 as a spin-out from the University of Wisconsin, University of Washington in Seattle. Our mission is to bring the power of bioengineered cell models to people developing therapies for the world's most devastating diseases. We knew this would be a challenge, since historically these approaches have involved very complex techniques that require a ton of expertise. And we approach this by making turnkey platforms that combine cells, systems, and data that represents complex human biology in vitro, but do not require expertise outside of basic laboratory skills. We use these systems to deliver data on how a potential therapy can influence these systems and drive decisions regarding the safety and efficacy of your compound before it goes into the clinic. I'd like to discuss a little bit more about how we do that now. So, as I mentioned, Curi Bio is accelerating the discovery of next-generation human medicines by integrating human cells, systems, and data into a paradigm that we call the Curi engine. We provide human cells, induced pluripotent stem cell derived cell lines, advanced 3D biosystems, and software solutions for analyzing the data. One of the things I really like about Curi Bio is that we have all of these systems integrated under one roof. And I like this because I think it provides a better experience for the scientists that we serve. There's no need to hassle with different vendors. I can remember one instance very early in my career, where I was first setting up an instrumentation imaging setup. And I had to try to synchronize my stimulator with my microscope. And when I contacted the microscope company, they told me to contact the stimulator company. And when I contacted the stimulator company, they asked me to contact the microscope company. And eventually I got it figured out, but it took a lot more effort than I really thought was needed. With Curi Bio, if there's something wrong with your EHTs, you know who to call. It's Curi Bio. But at the same time, even though we have this integrated system, it's also an open system. You don't have to use all of these components. You can use your own cells, for example, with our 3D biosystems and our analysis software. I'd like to take a closer look now at our 3D tissues. But first, I want to take a second to look at the state of the art otherwise. So if you were interested in muscle physiology and you wanted to measure something like force output, the alternatives until very recently were measuring contractile force with very invasive, expensive, and time-consuming animal models. Curi Bio is aimed to revolutionize sort of these approaches with our 3D technology. Our 3D technology is designed to sort of strike a middle ground between these invasive in vivo experiments and 2D experiments that lack physiological relevance. The culture system involves casting these 3D tissues between two supporting pillars. One is rigid and stays in place, and the second flexible post deforms under the contractile force of the tissue. And this sort of structure is very helpful in maturing these tissues because the sort of tension applied between the two posts helps the fibers align correctly and mature. In addition, this 3D system provides enhanced chemical and mechanical extracellular matrix cues. And these tissues are also formed from a heterogeneous cell population similar to what you might find in vivo. And these cell-to-cell interactions further improve maturation. Additionally, there's an extended culture time. So if you've worked with 2D muscle cells, you might be aware that they're only viable in culture for a short period of time, days to slightly over a week. 3D tissues, on the other hand, can be cultured for weeks to months or longer. And this culture time is necessary for the gene expression cascades to have time to run their course and to observe metabolical changes. Finally, this 3D structure is important for clinically relevant functional output of muscle. This 3D structure allows us to measure the force generated by the tissue, which is simply a very difficult task to do with 2D cultures. So I'd like to take a second now to look at the systems that we have for making measurements from these tissues. Our first flagship instrument was the manta ray platform, which is designed for stimulation and contractility measurements of these tissues. We also have Stingray, which is a dedicated stimulation instrument, which can either be used as a standalone instrument to provide a stimulus to enhance maturation of the tissues, or it can be combined with our Nautilai platform for providing stimulus during imaging. Our Nautilai platform is our fluorescence imaging platform designed for 2D and 3D measurements of voltage and calcium flux. And I'd like to take a closer look at the manta ray instrument now, because that provides inspiration to many of the experiments that we investigate. So as I mentioned, the tissues are formed between these two posts. We have one rigid post and one flexible post. Another key is that inside of this flexible post, there's an embedded magnet, and this allows for the manta ray system to track the motion of this post when the plate is placed on top of the manta ray, and provide simultaneous contractility output for all 24 tissues. But contractility, while it's often our measure of interest, is an incomplete picture. So one question we might want to ask is, if the tissues stop contracting, why did they stop contracting? So we built the Nautilai to investigate the underlying calcium and voltage signals so we can have a better understanding of the physiology in those cases. So the Nautilai instrument was designed with calcium imaging and voltage imaging in these tissues in mind, but we also designed it to be a flexible platform to also allow imaging on industry standard SBS compliant multi-well plates. We've tested it with 24, 96, and 384 well plates, but it may be compatible with others, but we haven't tested those yet. In addition to designing it with the application in mind, we've designed it with the user in mind, to be as simple of a system to use as possible. I'll show you later that you can actually acquire data in as few as 3 clicks with this system. The imaging platform was also designed to be compatible with our existing analysis software pipeline. This allows for fast and convenient analysis. After acquisition, the data can be uploaded to our Pulse cloud platform for automated analysis to extract parameters such as fluorescence transient amplitudes and frequencies. That's, I think, a good overview of our company platforms. I'd like to take a step back now and look at, talk about for a second why calcium imaging is difficult. Many of you have probably performed these types of experiments before. In my experience as well, optimizing staining is complicated, data acquisition is slow, and analysis is tedious. I'd like to take a second now to ask the audience, what do you all find to be the most challenging part of your calcium imaging experiments? All right, we've got a couple answers coming in. I see data acquisition and data analysis are very key challenges for everybody else as well. That's been my experience. I'm glad I didn't have to answer this poll. Honestly, it would have taken me all day to figure it out. Because in my experience, they're all challenging. Optimizing your staining protocol can be very complicated, especially if you don't have a very good starting point. It's also a little stressful because everything else is built upon this staining protocol. So if you make a mistake here, it sort of cascades into everything else. The one, I guess, point of grace, I guess, is that you only have to do it once at least. Once you optimize it, you can continue to use that protocol for years. But from day to day, you still have to do data acquisition and analysis, which will remain challenging even after the optimization has completed. Looking back at my career, the data acquisition was always challenging for me because we had an imaging platform from one vendor, simulation platform from another vendor. The microscope stage was probably made from somebody else. So we have several pieces of software to control all of these. Then when we do the acquisition, they all have to communicate properly to provide the data output. And then when we go to do it analysis, the data is, in several different file formats from several different manufacturers. And analysis, we always had to do a combination of ImageJ or something and some custom written R scripts or Python scripts, which were error prone and difficult to use. So those are the sort of pain points that we tried to address when designing Nautilai. Sorry, my slides don't seem to be advancing. Hopefully this resolves in a second. There we go. All right. So I'm going to take a second to take a brief overview of the workflow on Nautilai. First, you follow our optimized protocols for tissue production, maturation, and dye staining. You can sort of skip that optimization process. After the labeling, you can run your acquisition with as few as 3 clicks on the Nautilai software. Quickly upload that data to our Pulse automated analysis platform to extract amplitude, frequency, pulse width parameters, and more. And use that data to drive decisions about whether your therapy is safe and effective. I'm going to take a step back and talk about our labeling protocol now. I'm going to talk first about what our protocol looked like when we started. When we started, there were no real reliable protocols for EHTs, for labeling EHTs with calcium dyes. You could find reliable protocols for labeling HeLa cells or labeling cardiomyocytes in 2D or even labeling neurospheres or other preparations, but 3D engineered tissues are pretty new. So we started with the manufacturer's protocol for Fluo4, and I'm sure it worked pretty well for them. And in fact, I've used this protocol successfully without modification for imaging HeLa cells, but it didn't work very well for EHTs. As you can see in the graph here, the signal to noise was relatively low. The responses were pretty small, and we had significant photo bleaching. Additionally, this was before we really developed the Nautilai platform, so imaging was performed using Micromanager or a similar microscope management software, and it was analyzed in ImageJ and or with custom written Python and R scripts. I'd like to talk for a second or a couple minutes now about the process we went through for optimizing the staining protocol, and then we'll review the other steps. I want to take a second to point out that we're optimizing our signal to noise. My goal is not to maximize the signal to noise. And the reason for that is that all calcium sensors detect calcium by binding to it, and this prevents it from carrying out its normal physiological function. So to illustrate that, I have a figure from Erwin Nayer's paper in 1992 where he patched a cell first with a pipette containing dye, and we can monitor the calcium signals as the dye goes into the cell. And we can see at first the calcium transients are very large and fast, but as the dye enters the cell, the transients get smaller and slower. Afterwards, he repatched the cell with a pipette without dye so that the dye was removed from the cell by the pipette. And we can see as the dye is removed, the calcium signals recover, becoming larger and faster. Oftentimes, the dynamics of calcium aren't really what we're concerned about. I view this as a way to quantify the changes that we've made to our physiological system. So they're just a symptom of a larger problem. Additional physiological effects include inhibition of neurotransmitter release, protection from ischemic injury, and disruption of activity-induced gene expression. With that being said, let's turn our attention to maximizing your signal while minimizing these physiological changes. The first consideration is to use a dye with an appropriate affinity. So dyes vary significantly in how tightly they bind to calcium. If the affinity is too high, the dye will be bound to calcium at rest and you won't see very large changes. If the affinity is too low, then the fluorescence at rest will be too small to quantify and you won't be able to accurately measure changes. So what we want is a dye that's just right. So let's take a closer look at this one because it's an important factor. So the dissociation constant, or KD, is a measure of the dye's affinity. Sort of importantly, the KD is available from the manufacturer of the dye, so you don't have to worry too much about the exact biophysics of what it means. You can just look up the parameter. And typically a dye with a KD near the physiological calcium range that you want to measure is typically a good choice. So in the first example, this is a response from a neuroendocrine cell. We can see that green BAPTA1, which is a high affinity dye, produces a very small fluorescence change in response to stimulus delivered by a patch clamp. Calbright 630, which is a lower affinity dye, on the other hand, provides robust signal changes of approximately 300% in response to the same stimuli in the same cell. I want to turn our attention now to these experiments that we did for optimizing the protocols for EHTs. I knew, based on the physiology, that the resting calcium is around 100 nanomolar, and the peak calcium concentration is around 1 micromolar. So based on that, Cal 520 and Calbright 520 would be good choices. So we started our optimization protocols from that. And we can see that where we ended up with Calbright 520, which is the lower affinity of the two, ends up being the better choice. I want to expand a little bit on the KD. So given the choice between two dyes that perform similarly, we should generally prefer dyes with the lower affinity. And this is because lower affinity dyes have a smaller impact on calcium dynamics, making them a better choice when possible. So on here, what we're showing is a computer simulation of a hypothetical cell that has a transient with a fluorescence increase and then a subsequent fluorescence decay rate. And the baseline condition for this was a 50 millisecond decay rate. And then we ran the simulation without dye, where it was a 50 millisecond decay rate, and then with a fixed concentration of dye at different affinities. And we can see that high affinity dyes have a significantly larger effect, and lower affinity dyes produce much smaller perturbations to the physiological system. It's important to note that these effects are much larger for small KDs, and for large KDs, they become negligible. Another consideration is that we should prefer dyes with a large dynamic range. So the dynamic range refers to how much the fluorescence increases when the dye binds to calcium. A dynamic range with a dye with a dynamic range of 10 gets 10 times brighter when it binds calcium. So if we consider to the very extreme low point, consider a dye with a dynamic range of 1%. If it only gets 1% brighter when bound to calcium, the changes in fluorescence will be very small. And the dynamic range dictates how much sensitivity you get to calcium changes. So when we're looking at a drug-induced change in fluorescence amplitude, high sensitivity is very important for these types of experiments. Another consideration is to use the lowest possible concentration of dye. So simply put, more dye means that you will have larger physiological changes. The minimum dye concentration you can use is really limited by your hardware and the software analysis. So on the hardware side, you want something with very low signal to noise, like our Nautilai platform. And on the analysis side, if you're doing higher throughput, you want to do automated analysis, you either need very good algorithms or data with a good signal to noise ratio. Fortunately, Nautilai provides both. Another consideration is using reagents and media and consumable optimized for fluorescence. So we're looking for changes in the brightness of the calcium dye. We're not interested in the fluorescence of the 24-well plate or the media, etc. It's good practice to subtract the background data, and Nautilai makes this easy to do with the click of a button, but it's best practice to minimize the background in the experiment to begin with. And this is, the reason this is because you can subtract the average background from your recording, but you can't subtract the background noise. So if you have very bright background, this significantly reduces your signal to noise. And in the worst cases, that can be a fatal flaw in the experiment. The final consideration is we should prefer to use the brightest dye possible. So for example, Fluo4 and Cal 520 both have a KD of around 300 nanomolar, so they're sort of medium affinity dyes. And they both have very large dynamic ranges, but Cal 520 is 5 times brighter. And this sounds like a pretty big change, and it is, but it actually underestimates how big of a result this has. Because the expected, this leads to a 94% decrease in the expected production of reactive oxygen species, and therefore a 94% reduction in photo bleaching and photo damage caused by the imaging. And I want to make one final note about getting started with our protocol. So we recommend getting started with Cal 520 because it's a really fantastic dye. And when getting started, we recommend that you follow the protocol exactly. This is because small changes can have really big effects. For example, when Cal 520 is dissolved in a DMSO pluronic F127 solution to make a concentrated dye loading solution, the dye is incorporated into pluronic F127 micelles, and these micelles remain stable when diluted in media, and this leads to efficient dye loading. However, if you dissolve the pluronic F127 and DMSO directly in the media, and then try to dissolve the dye in that, instead of forming micelles, we have single F127 monomers, and the dye tends to form insoluble aggregates and fall out of solution, leading to very poor dye labeling. So after you have a working protocol adhering as closely as possible to our optimized protocol, then you can adjust the protocol for optimal performance or for convenience by changing factors such as dye concentration, loading time, temperature, and media conditions. Now that we have our starting protocol, let's consider the rest of the workflow. After loading, we can place the plate directly into the Nautilai for imaging, and then proceed with the automated analysis. And afterwards, we will make some decisions based on the data. So when you place the plate in the Nautilai controller and start it up, the software interface looks exactly like this. And there are only a couple of parameters here, but they're all that you really need to do your calcium imaging experiments. So I want to share quickly just how easy an acquisition can be. So first you select the plate format. Here we're doing imaging with Curi Bio manta ray mini tissues. And then you can use start live acquisition, which updates the display, and then you start the acquisition. And using our optimized protocols, the default parameters produce really good recordings. So that being said, let's let Nautilai do the heavy lifting for a second and make some measurements. So after you click Start Acquisition, the rest of the process is fairly automated. The acquisition proceeds through the different stages to acquire data from the entire plate. And it'll continue going for a couple minutes, but we don't really need to watch that. So let's look at what happens next. So after the acquisition is completed, if you go to the data directory, you'll find the raw data as well as a video that can be quickly viewed with your favorite media viewer. Additionally, the software will automatically generate regions of interest based on the plate format that you selected. And based on these regions of interest, it calculates fluorescence versus time recordings, and it saves all of these in a sort of quick, easy to view format that you can double check to make sure that your experiments are going as planned. Then we can take this data and upload it to the Pulse Cloud platform. For automatic extraction of the detailed kinetics from every recording. So Pulse will automatically calculate key parameters such as the peak amplitude and the period. And it will provide you with a convenient, easy to use Excel format, which with plots illustrating the peaks that were detected and the values, and summary statistics of all of our key metrics. Finally, I want to reiterate that this platform is compatible not only with our 3D manta ray tissues, but also it's compatible with conventional SBS compliant, with conventional culture and SBS compliant multi-well plates. And we've tested this in 24 well, 96 well, and 384 well plates. So now that we have our data, let's take a second to talk about how to interpret this data. There are generally two ways we can look at calcium imaging. We can approach calcium imaging. We can perform quantitative imaging or semi-quantitative imaging. So by semi-quantitative imaging, I mean we're looking at relative changes in fluorescence amplitude. So this will tell us that calcium increased by 10%, for example. Quantitative imaging, on the other hand, looks at exact calcium concentration values. So if we want to know that the resting calcium concentration in the cell was 50 nanomolars, that's a quantitative calcium measurement. We might be interested in these measurements if we were looking at a disease model such as DMD or Alzheimer's, where it's been established that resting calcium concentrations are changed in these disease models. However, these measurements are very difficult to make. So to illustrate this, I want to take a quick look at this figure from the Thomas et al. Paper from 2000, where they evaluated several different calcium dyes in HeLa cells. And one thing that I would like to point out, or a couple things that I'll point out, is that the HeLa cells at rest have a non-uniform staining pattern. So they're much brighter in the nucleus here for some dyes, although other dyes it's pretty consistent, with lower staining in the cytoplasm. Now at rest, we don't expect actually for there to be a difference in calcium concentration between the nucleus and the cytoplasm. So what this indicates is actually that the dye is behaving differently in those two areas. Additionally, the dye is compartmentalized into subcellular compartments such as the mitochondria and the endoplasmic reticulum. And the end result is that the fluorescence that you measure is a combination of fluorescence from these different cellular compartments, which all behave differently and have different calcium concentrations, which makes it very difficult or impossible with AM esters to make quantitative calcium measurements. One final challenge is even after we addressed those, the calcium concentration can only be calculated at equilibrium. So that's down here where the changes are not very large. And arguably this is the least interesting part of our calcium imaging experiments. So I want to take a step back and sort of tie that all together now. This really hurts me to say because I spent the last 15 years trying to develop better methods for quantitative calcium imaging. But the truth is calcium concentrations can only be calculated in very well-defined and well-controlled experiments. For example, if you patch clamp a cell with a pipette to introduce the dye so that it's only in the cytoplasm and the nucleus, and then you have a very high resolution imaging set up so that you can ignore the nucleus where the dye behaves differently and only get data from the cytoplasm. Then, under certain conditions still, you can calculate calcium concentrations. In most situations, absolute calcium can't be calculated, and changes should be interpreted as relative changes in calcium, as, for example, fluorescence over F0. But this is still a really powerful technique. So I want to take an overview of our optimized imaging protocol to sort of tie this all together. So we started with the fluo4 labeling protocol from the manufacturer. And under these conditions, labeling in our engineered tissues was very poor. A lot of this was likely due to the formation of fluo4 aggregates instead of the fluo4 labeled in the cells. So the labeling efficiency was very low, requiring us to use a very high LED intensity to get high fluorescence signals. This leads overall to small changes and rapid photo bleaching and photo damage. Additionally, when we were starting, we had this very manual process using Micromanager for acquisition and ImageJ for analysis. And today, we have this optimized labeling protocol with CalBrite 520 for EHTs. A significant portion of this improvement is due to the labeling protocol being more efficient at delivering dyes, but in addition, Cal 520 is a brighter dye to fluo4, which provides further improvements. The automated acquisition and analysis workflows result in these experiments being much easier to do than I was sort of used to from my experience. So, for example, the optimizing protocols for calcium imaging when I was in graduate school probably took me a couple of weeks. That was patch clamping individual cells, so it's not a direct comparison, but it's still a significant contrast to optimizing this protocol, which took about 2 hours with our automated workflows. That's including time for 1 hour for the incubation and imaging and analysis. So now that we've optimized our protocol and acquired some data and thought a little bit about how we can interpret it, I'd like to turn our attention now to some example applications with illustrations on Nautilai. So the first application I want to talk about is looking at peak calcium concentration versus stimulus frequency. This is an important application because there are several disease models where the deficits become more apparent and exaggerated at higher stimulus frequencies, but also calcium and force summation during higher frequency stimulus is a hallmark of tissue maturity. So in vivo, as you increase the stimulus frequency, you will see an increase in calcium concentration summation and force summation. When you use young immature tissues, such as these tissues here, what we find is a sort of a flat curve or a flat line where the calcium concentrations don't increase at higher stimulus frequencies. As these tissue matures, we would see that sort of the expected in vivo result for skeletal muscle tissues. For heart tissues, on the other hand, the state of the art is a little bit different. It's well documented in the literature that the EHTs don't fully mature yet with how they're being made. So this is a problem that we are actively working on, and we made some recent progress. I want to share a little bit of that data here. So in this maturation protocol that we tried, these tissues were cultivated first for 21 days, and then a chronic pacing protocol was applied starting at day 21 for 2 weeks. And then these data were acquired on day 58. So similar to skeletal muscles, heart muscle in vivo should have a positive relationship. As you increase the stimulus frequency, we would expect to see an increase in force and an increase in fluorescence. But in these 3D cultured tissues in both wild type and DMD models, instead we see a decreasing force and decreasing fluorescence changes with higher stimulation frequencies. If these tissues are matured by applying chronic stimulation, we don't see a fully mature phenotype, but we see an improvement where these changes are significantly less pronounced. We're going to take another look at another experiment we can do with our DMD disease model. We would like to know generally what are going to be our most sensitive parameters when performing comparisons for therapeutic modes, for example. So in some pilot experiments we did with our DMD disease model, we examined the calcium, the fluorescence dynamics in wild type versus DMD disease model. We can see a sort of striking difference in the waveform shape, and these differences are most sensitive in the decay time from peak to 10%. We see significant differences in our disease model and wild type, and also in the decay from peak to 90%. Another potential application is using this for acute drug effects, such as the example illustrated here for verapamil. Verapamil is a voltage-gated calcium channel and voltage-gated potassium channel blocker. It's used for the treatment of high blood pressure and tachycardia, and with more than 2 million prescriptions per year in the United States, it's generally considered very safe and very effective and has been designated by the World Health Organization as an essential medicine. It's also worth noting here that verapamil is known to be a false positive on in vitro hERG activity screens. So this makes verapamil a compound of significant interest for developing improved screens for that type of, for hERG activity. In addition to acute ion channel blockers, we can look at compounds such as isoproterenol, which is a medication used to treat bradycardia. Isoproterenol is a non-selective beta adrenoreceptor agonist, and those receptors are from the GPCR receptor superfamily, which has a fairly complicated signaling cascade, but the end result is that stimulating that signaling pathway produces IP3, which binds to IP3 receptors on the endoplasmic reticulum, stimulating calcium release, which produces an increase in oscillation frequency and an increase in oscillation magnitude. I'd also like to take a look at one of our upcoming physiological models. So we have recently made significant progress on developing a model for the neuromuscular junction. Developing a model for this in vitro has been very challenging for the field as a whole. But this is sort of fundamental. It plays a fundamental role in human health, and deficits in this system can lead to devastating diseases. So I'd like to take a second to talk about how we built this model on top of our 3D skeletal muscle culture system. So it starts with a consumable, which is a modified version of how we normally make these 3D skeletal muscles. In this modified version, there are these sort of semi-spherical indentations at the bottom, where neurospheres can come to rest. After those neurospheres have been seated in this casting plate, a hydrogel is placed over those neurons, and then it is bonded to a mature skeletal muscle tissue to produce this skeletal muscle and motor neuron co-culture. After we allow them to mature, we can take a look at this system under a microscope with immunohistochemistry staining, and we observe these punctate localizations of tubulin from the motor neurons, projections co-localizing near the myosin heavy chain labeled skeletal muscles. In addition to these structural hallmarks of maturity, the NMJs this model produces functional NMJs. This is illustrated by this experiment where where the NMJ co-cultures were stimulated with a blue light. So one important point that I forgot to make was that the neurons contain the blue light sensitive opsin channel rhodopsin 2, so that when they're exposed to blue light, the neurons increase their firing frequency, but the muscles have no response to the blue light. And we show robust contractile forces generated in this model in response to blue light. For calcium imaging, unfortunately, the blue light produces large imaging artifacts. But we're still exploring this in this measurement methodology for calcium imaging to observe the physiology sort of in a potentially higher throughput format. So one thing that we can notice is that in the NMJ co-cultures, we find some ROIs that are highly active and some ROIs where there is no significant activity. And there will be other ROIs that are active but not at the same frequency or at the same phase. So when we averaged over the whole tissue, we don't really see a very large signal. But if we choose our ROIs more appropriately, we can see some spontaneous activity in this NMJ system. In comparison, the control tissues have no activity really, regardless of the ROI you choose. And one final potential application, not related to calcium imaging, is voltage imaging on Nautilai. So in this experiment, we used fluovolt to label the tissues and measured the spontaneous oscillations in voltage in our EHTs. And we can see very nice spontaneous oscillations in voltage using this model. Sorry, the slide's taking just a second to advance. There we go. It's worth noting that these changes are much smaller than the calcium changes that I showed before. In the videos I showed previously, the calcium changes were pretty visible, even just watching the video. If you look very closely, you can see some of those changes here in the video. And we can see in the graphs the changes clearly, but at a much smaller amplitude. And this is sort of inherent to the voltage imaging method because the calcium dye is in the cytoplasm and the voltage dye is in the plasma membrane. There's simply much less voltage dye, so the signals are smaller, but still enough to form a very useful assay. This is an application we're still actively pursuing, and we hope to have an application note or something like that in the next coming months to share with you all. With that, I'd like to thank you for your time and thank you for your attention. Please feel free to reach out with any questions. We'll open this to Q&A, but if we don't get to all of your questions, please do feel free to reach out to me directly.
Okay, perfect. Thank you, Shane, so much for that wonderful presentation. That does bring us to our Q&A portion. Everyone can continue to submit questions, but we have received a few already, so I'm going to start with those. Okay, first question. Does Nautilai have stimulation capabilities built in? If not, what stimulation instruments have you used?
Thanks for that question. We have the stimulation capability is not built in, but we have a stimulator. We have our standalone Stingray, which we've used successfully with Nautilai, and we've also used our manta ray instrument to provide stimulation for Nautilai.
Okay, perfect. Thank you. Second question. Is the Nautilai instrument environmentally controlled for long-term studies?
Yeah, thank you for that question. Yes, it is. We have temperature control as standard, as comes as in the standard model, which allows you for doing by itself sort of medium-term experiments. And we also have a CO2 and humidity controller, which is an optional add-on, which allows for very long-term recordings. We haven't explored that to the greatest extent. We've performed experiments for up to 24 hours in that chamber, but in the literature, it's been reported that these types of stage-stop incubators can be used for experiments for days to up to a week or so.
Wonderful. What is the maximum acquisition speed of the instrument?
The maximum acquisition speed of the instrument is 250 frames per second.
Awesome. Can I use any plate format with Nautilai, or is the platform limited to specific plate manufacturers?
You can use almost any plate format you want, with the caveat that the instrument needs a configuration file for every plate format. So if you're using an exotic plate format that we've never seen, we might need to make a configuration file for that. But otherwise, I don't know of any plate formats that can't be used.
Perfect. Next question is, have you used Nautilai with other 3D organoids?
We've done some very preliminary experiments with neurospheres, but the bulk of our 3Ds have been with these engineered tissues.
Perfect. Next question is from Austin Berry. Do you feel there is an advantage of using a radiometric dye like Fura 2 AM over non-radiometric dyes like Fluo 4 AM or vice versa?
In certain cases, there are. It depends on the experimental design and your hardware. So when we were talking about the labeling protocol, I mentioned that the dye goes into the cytoplasm, it's in the nucleoplasm, in the endoplasmic reticulum, and in mitochondria, and it has different characteristics in all of those compartments. So Fura doesn't really correct for that deficit. It will correct for things like variation in LED intensity, and to a certain extent, variations in labeling intensity. But it can't correct for the dye being in different locations. And in these, we also have different cell types. So there's fibroblasts and cardiomyocytes. And the dye probably has different properties in both of those. And almost certainly both of those cell types have different calcium handling properties. So the calcium concentrations in those cells will be different, making it very difficult to get more useful information from those types of ratiometric dyes in these types of systems.
Okay, thank you. Next question is from Martin Morrow. How robust are 384 well data with Nautilai, and how strong is the correlation between data from 384 well plates and those with fewer wells?
As far as robustness, in our hands, the largest source of error for 384 well plates has been our robot protocols for plating and dispensing liquids. So the monolayers, since there's a much smaller area, end up being more fragile. So you have to be more careful to make sure you don't accidentally start peeling off those monolayers, for example. In addition, the smaller area of the plates, these smaller wells, have slightly different physiological properties. So the resting frequency in monolayers from 384 well plates seems different from 96 well plates. We haven't looked at this in detail. I'm not sure if it's a chemistry thing. Maybe the plate manufacturer is different was part of it, or if it's a effect of the surface area because the calcium waves take longer to propagate across the well. So maybe there's a sort of sensing mechanism there to keep it synchronized so that the beat rate has to be slower in the larger ones for the wave to propagate entirely. I should point out I'm speculating here. I don't know for sure why, but we do observe some differences. I think that was a two-part question and I answered one part. Can you remind me what the second part was?
Yeah, how strong is the correlation between data from 3 to 4 well plates and those with fewer wells?
Okay. So with those caveats that the resting oscillation frequency seems to be different, drug effects seem to be well correlated. So isoproterenol still increases frequency in both. We don't have enough data to have a very tight range on the effect sizes. So I don't know that it's the same size of effect, but broadly speaking, we see the same types of effects.
Perfect. Next question is from Gia Lee. How many cells, muscle fibers, cells/muscle fibers are in the 3D EMT bundle?
That's a very good question, and I really should know the answer to that, but honestly, I don't know. So we will have to get back to you on that one.
Yeah, we can get back. Also from Gia, will all cells be scanned simultaneously?
In the Nautilai instrument, it images 1/6 of the plate at a time. So for a 96 well plate, it's imaging 16. And for a 384 well plate, it would be imaging 64 at a time.
Perfect. Okay, so next question from Melanie is kind of four-part question or 4 different questions. So we're gonna take it in chunks. Can you do all types of stimulation protocols, or are there some limitations?
All types of stimulation protocols. I can speak, and I'm not entirely sure what that question means, so I can speak sort of in generalities of what we can do. So the Stingray and Manta Ray both provide stimulation in a current-controlled method. And so it's not rather, compared to voltage control, for example. So you specify the current that will be delivered. And you can specify different pulse train frequencies, and these can be specified on a well-by-well basis as well.
Okay. I think that's pretty solid for all of our questions. Yeah, okay, fantastic. Thank you, Shane, for all of those answers, and thank you everyone for sending in all your questions. We're gonna go ahead and end our Q&A portion now. If we did not get to answer your question here, we will follow up with you via email afterwards, or please do feel free to reach out to us at contact@curibio.com or directly to Shane's email listed on the screen. And we definitely want to make sure that your questions get answers. A big thank you to everyone for participating in today's webinar. You will be receiving a follow-up email from us regardless, with access to a recording of this event. And then, of course, another big thank you to our speaker, Dr. Shane McMahon, for that wonderful presentation. We hope you found this webinar informative. Have a great day, everyone.